So our presentation is going to go over the beginning of the cell cycle, and I have the overview in section 12.1. So the overview. Rudolf Virchow in the 1800s summarized the life in relation to cells as the Latin phrase omnis cellula e cellula, which translates to every cell from a cell. And this really illustrates how life is brought about and continued by the cell division process so that life can continue. The importance of the cell division process is really exemplified when you look at a unicellular organisms, because when that divides, it creates and it gives birth to a whole new cell. So the cell division process is not just a trivial process, it's something that can give life to an entire new organism. And it also allows multicellular organisms to expand and develop. And it is not only for the growth, but after the organism has fully matured, it's helpful by repairing the damaged cells and renewing old cells, and then replacing them once they have died. So the cell process overall is imperative to the continuity of life, and it's crucial to the life cycle of both a cell and an organism. And the cell cycle entails from the birth of a cell to the division of the one cell into two daughter cells after its DNA has been replicated and such. An important part of this process is passing the DNA onto two cells, and that starts with the organization of the cellular genetic material. So the cell division usually entails the copying and distributing of its entire genome, which is a complete set of the genetic material present in a cell. So it has to copy the entire DNA molecules before it can divide into two daughter cells. Just a fact for reference, the DNA in a cell is greater than about 250,000 times the length of the cell diameter. Some vocab. Chromosomes are thread-like structures of nucleic acids and protein found in the nucleus of living cell. They carry the genetic information in the form of genes, and this is what the DNA is stored in, like a chromosome. And before a cell can divide, we've been over this, it must copy the DNA, which is packed into these chromosomes. Okay. Does anybody have any questions? So, what is the DNA packed into, Maggie?
Chromosomes.
Yes. And Cameron, what must the cell do before it can divide?
Replicate the genetic material.
You got it. Okay. Cellular organization of the genome. So the chromosomes contain the extensive, vast amount of DNA that codes for specific proteins production. So, in short, it specifies the inherited traits that the cell or the organism as a whole gets from its procreators. Chromatin, another vocab word. It is the entire complex of DNA and proteins that is the building material of chromosomes. So basically it goes DNA and then genes, and it's stored in chromatin, which is in a chromosome. Every eukaryotic species has a specific number of chromosomes. For humans, it's we have two sets of 23, which makes that. Anybody? Al?
46.
There you go. Awesome. So another vocab word is a somatic cell, which is all the body cells except for the reproductive cells, which in humans are sperm and eggs. And this contains 46 chromosomes, two sets of 23. Each set is inherited from a parent. Does anybody have any questions so far? Moving on. And one more vocab word is a gamete, which is just a sperm or egg cell in humans, and it has 1/2 of the total chromosome count that's going to be passed on to a new organism once it unites with the opposite gender gamete. So it has a single set of 23 chromosomes. Okay. Does anybody have any questions? No?
So for the distribution of chromosomes, when a cell is not dividing, and even when it's just like replicating the DNA for division and preparation of division, the chromosome is in the form of chromatin, which is inside the cell. And like Jacqueline said, chromatin is basically a thin fibrous strands made up of DNA, RNA, and nuclear proteins that make up chromosomes.
Clark, what does the word chromatin and chromosomes actually mean?
I have the word.
Here.
Yeah. Chromatin is like an entire complex of DNA and proteins that is the building material of chromosomes.
Right, but chromatin, the word chromatin is actually
Oh,
a word that
chrome
means
means
something.
color, and
Right.
then some means body, I think.
So, all right, for
Wait, for
chromosomes, so
Latin?
colored bodies on the slide, right?
Yeah.
Like,
And
that's
it's called
originally what they look like on the microscope
that.
slide.
Yeah, it's
little-
called that because it like picks up light when they like die and stuff.
Yeah, you can pick up a chromosome on a microscope, but you can't see chromatin on a microscope because they're thin.
Right, but chromatin actually appears as just like thin little threads.
Yeah.
And that's literally what the word chromatin means, is colored threads.
Yeah, and chromosome is colored body. Yeah.
Okay. And after a cell has replicated the DNA, the chromosomes condense. The chromatin condenses into a chromosome, and then each chromatin fiber becomes densely coiled and folded. And here, so this is what chromatin would look like inside of a cell, opposed to a chromosome being a thicker and more dense body inside of the cell. And so after each chromosome has duplicated, it becomes two sister chromatids. And a sister chromatid is, they both contain identical DNA molecules, and they are attached along their lengths by adhesive protein complexes called cohesions. Jack,
You said chromatin condenses
Yes.
into what?
chromosomes.
And how does it do that again?
It coils and folds within the cell, like after all the DNA has been duplicated,
Oh, okay. I was wondering.
then it changes from chromatin to chromosomes.
And that's why it's like.
It's sort of like if you're going to pack up the library to make another library, you're gonna have to, wrap up all the books and get them in a way of being able to move them. That's kind of like if you're gonna you prepare the cell for division, you're gonna have to condense all that chromatin into chromosomes so that it can be properly moved. Now, what is that central constriction point on the chromosome called?
Oh, we get to that.
Okay.
Okay. And trivia question: cohesion between two sister chromatids is most likely referred to as. Anyone know? Sam?
Oh, cohesion?
No. Yeah, sister chromatin cohesion instead of just cohesion. Alright, so later in the cell division process, the two sister chromatids of each duplicated chromosome will separate and move into two new nuclei, forming one at each end of the cell. And true or false, the sister chromatids that have been separated are now considered individual chromosomes. John?
True.
Yes, they are now, once they've separated, they're each their own individual chromosome. Oops. Alright, because the sister chromatids have split, they're now considered individual chromosomes, and each nucleus receives a collection of chromosomes that are identical to the parent cell. Okay, so in review, we have we start out with an individual chromosome, and then it's separated, the DNA is duplicated, and we have two sister chromatids that are attached along their length with the sister chromatid cohesion. And in the middle, like Doc said earlier, this central area, they're attached at a region called the centromere. And after this, They are then separated into individual chromosomes, and each are going to a different nucleus.
Okay, so now we're gonna talk about interphase, which is kind of the first step before we actually get to mitosis. And it counts for 90% of the cell cycle, and it's the part of the cell's life when it's just growing, and since it's growing, that means it's alive, but it's not dividing. So during this phase, the chromatin is enclosed in the nucleus, and the nucleus has one or more nuclei, and there are two microtubule organizing centers. These are the centrosomes in animals. And there are three stages that make up interphase, and they are the G1 phase, the S phase, and the G2 phase. So here we have a picture, and it shows the interphase, which is the green arrow. So you can see it takes up most of the cell's life, and a large chunk of the cell's life is spent in the G1 phase and the S phase. And then a very small portion is spent in the mitotic phase, which is just the dividing of the cell. So the first stage is the G1 phase. The G phases were misnamed gaps because at first it was thought that during these phases nothing was happening and the cell was inactive. But it was later found out that the G1 phase is actually a period of intense growth and biochemical activity. So because of this, it was called the G1 phase, and the G is named for growth. The second phase is the S stage, and it's very important because it is the synthesis stage. And during this stage, this is when all the DNA is duplicated in the nucleus. And Jacqueline kind of talked about that the DNA was duplicated, but can anyone tell me why it would be a good thing that we're duplicating the DNA? Sam?
So that it's ready once it splits.
Yeah, so since you have two sets of identical DNA, the one set can go to one cell and the other cell can go to and the other set can go to another cell in mitosis. So the other thing that is duplicated during this stage is the centrosomes, and these are the microtubule organizing centers. Can anyone tell me why it would be a good thing that the centrosomes duplicate? Think about what the microtubules do, and if you had the section on mitosis, you might understand that the microtubules are a big part of mitosis. Sam?
So that there's one for each cell, once it's divided.
Yeah. So the centrosomes are really important because they form the microtubules, which will start to pull apart the two the chromosomes at the center of the cell, and that's how you get your two separate cells in mitosis. The last stage is the G2 stage, and this is the second growth cycle of the cell. The cell completes its preparations for division, and at this point the two centrosomes begin to separate from each other and they start moving to opposite poles in the cell.
So before we can talk about mitotic spindle and its role in the cell cycle, we kind of have to review. If you guys remember from chapter 6, a microtubule is a hollow rod that's made from the globular protein tubulin, and you can find it in the cytoplasm of eukaryotic cells, and what it does is basically it supports and it shapes the cell. And so what the mitotic spindle is, it consists of fibers of these microtubules and associated proteins, and you can find this in the cytoplasm during prophase. The cytoplasm itself, it's going to partly disassemble so that they could provide microtubules to form the spindle. And these, the microtubules are going to elongate so that more, the microtubules will elongate as more tubulin is added, making these strands longer. So to kind of give you some definitions here, a centrosome, it's an organelle that will organize the cell's microtubules. It's also referred to sometimes as a microtubule organizing center. And this is where the assembly of the spindle microtubules begins. And so then centrioles, you find them they're at the center of the centrosome, and they are present in animal cells. They're not always present in plant cells, but they're not exactly essential to the cell division process. And so in interphase, the single centrosomes are going to replicate, and they're going to go they're going to go forming two of them. And what's going to happen is during prophase and prometaphase, they're going to move to opposite sides of the cell. and now that they're on opposite poles, they're gonna form asters, and they're little extensions off of the centrosomes. And so the whole spindle itself is gonna include the centrosomes, the spindle microtubules, and the asters. So to kind of really explain what that is, so these are the centrosomes that are on opposite sides of the cell, and then inside you can see the centrioles. These long strands right here, those are gonna be the spindle microtubules, and then you see these little projections off the end. Like this is the outside of the cell right here, these little projections off the end would be your asters. So then, what is a kinnicature? So these are structures of proteins that they relate to specific sections of DNA. And so each sister chromatid has one of these, and both kinnicatures are gonna face each other. And so in prometaphase, the spindle microtubules are gonna attach to these kinnicatures, and they're gonna form what's called kinnicature molecules. And so what's gonna happen is, so when these first begin to form, like one side is gonna attach to this molecule, and it's gonna start pulling the kinnicature towards one end of the pole. But then the spindle molecule from the other side is gonna come in, and as they refer to it, it's like a tug of war until the kinnicatures can land in the middle on the metaphase plate because of the pull from the two spindle microtubules. And so so then by metaphase, the kinnicatures are gonna be aligned in the center on something called the metaphase plate. And so at this point, the spindle is gonna become complete because microtubules that, where they're gonna attach to other molecules that aren't kinnicatures, and they're gonna overlap and interact with them. And then the asters, they're gonna grow and they're gonna now attach to the plasma membrane.
So what's the purpose of the esters, or astral rays?
Well, I think it's because, I was just saying, so like, now they're gonna be attached to like the end of the plasma membrane, so like for support, like as the cell separates so that they stay connected. Alright, so then just like to give you a picture, so here the centrioles are still in prophase, they're still going to opposite sides of the cell. And then by prometaphase, you can see like it's starting to form the spindle microtubules, and it's just starting to attach to these kinnicatures. And then by metaphase, you can see that they've aligned in the center, and that the kinnicatures are attached by both sides. And so in anaphase, the chromatids are gonna split, and so there's two possibilities as to why this might happen. One possibility is that like the chromosomes, the microtubules are going to shorten, and so as a result of shortening, this pole is gonna drive the chromosomes apart to opposite sides. But another theory, and this one has like a lot of experiments that have proven this, is that there are motor proteins on the kinnicatures that are gonna almost travel along these spindle microtubules. And so the microtubules that aren't attached to kinnicatures or non-kinnicature microtubules, they're gonna be responsible for making the cell longer so that like it's big enough to separate. And so eventually what's gonna happen is the spindle is gonna disassemble, and because it's gonna polymerize like the different kinnicature ends, and cytokinesis will begin. So again, here they're being pulled apart. The polymers that it's disassembling,